Hi. So remember a long time ago when we learned about the cell theory, and we said that cells are the basic unit of all things, and all living things are made up of cells, and that cells come from pre-existing cells. Cells come from pre-existing cells. So today we're going to start talking about how that all happens. So cells come from pre-existing cells. So if you have one cell and you want to turn that into two cells, there are a lot of steps that need to happen in order for that to occur. And so the major thing that's going to be important that the cell transfers from this one cell into two cells is going to be its DNA. Because the DNA is like the instruction book. It tells the cell what to do, how to do it, and when to do it. And so getting the DNA duplicated, right? So first it's going to have to replicate the DNA, and then it's going to have to sort and divide so that all the cells have the appropriate information. So replicating the DNA we talked about before in previous unit as well. Helicase splits the DNA apart. Primase is going to lay down the primer. DNA polymerase is going to lay down extra DNA, like the DNA nucleotides, ligase. We have the Okazaki fragments. So all of that stuff, like we know how all of that works. So we're going to replicate our DNA so that we have two copies of it. Sorting and keeping everything organized then means that we're going to have to kind of pack all that DNA together, just like when you're moving from your house off to college, or just from one house to another house, you take all of your stuff and you're going to put it into boxes. You're going to pack everything up so that it's easier to move. It'd be silly to move individual dishes from your kitchen to the moving truck and then back into your new house. It'd be much easier if you put them all into a box, took that one box, put it into the truck, and then into the new kitchen. So that's what's going to happen here. Okay, so let's add a picture.
I have lemon juice.
Okay, have a lemon juice. Yes.
What?
Okay, have lemon juice. That's fine. Okay, so we have DNA here in the double helix, as we see, know, and love, right? So here's our DNA in the double helix, and so when it starts to condense, right, what's going to happen is the DNA is going to wind around histones. Now, these are just little proteins. So when it's nice and thin, when it's just pretty much the DNA and histones, it's very thin, and that's known as chromatin. Nice and easy, right? Chromatin, thin. And this is invisible under the microscope. So when it's so thin that we can't see it under the microscope. But it's going to start condensing, right? So those DNA wound around histones, they'll kind of wind around each other, and they'll create what are called nucleosomes. Nucleosomes will create coils. Coils will coil upon each other, making supercoils. And then eventually everything is going to condense into this huge chromosome, okay? Now, we call this, whenever you see, condensed DNA, so thin DNA is chromatin, condensed DNA is going to be the chromosome, okay? So thin DNA, chromatin, thin chromatin, condensed DNA is going to be the chromosome. So anytime you see the X-shaped pattern right there, that's going to be your chromosome. Okay, so this is like the box. It's filled up with everything inside. And so when we're when we're actually looking at an actual chromosome, we see that it looks like the X shape there, but it's really made up of two sister chromatids. Okay, so when we draw an X, it's almost like this coming together. Okay, so each little strand, one side of the X, is going to be a sister chromatid, which is one copy of the DNA. Remember how we replicated it? So this is going to be one copy, and then the other sister chromatid on the other side is the other copy. Okay, so they're going to be held together in the middle, as we'll see here, by a centromere. And so oftentimes I call this a duplicated chromosome. Okay, so a duplicated chromosome because it consists of two identical copies of the same information. Okay, now in this whole business of going from one cell to two cells, right? In this cell here, we're going to take all of the DNA and we're going to make two copies of it. So just like we see in this stage right here, we go from 1 to 2. And then they're going to be held together, right? And they're going to do all of the dance of mitosis, right? So there'll be a lot of moving around and organizing, and we'll get to that in a second too. But eventually they're going to separate so that one strand of each can be in the new cells, okay? So the tricky part here is that when the two sister chromatids are together, they make up one single chromosome, a duplicated chromosome. But when they separate, just like we see here, they are called single chromosomes. Okay? So I know that gets tricky. That's an O. Single chromosomes. So we have a duplicated chromosome, okay, and then made up of two sister chromatids. But when the sister chromatids separate, they become individual single chromosomes, okay? Duplicated chromosome, two single chromosomes. Duplicated chromosomes made up of sister chromatids, and then they become single chromosomes, which were the sister chromatids before, okay? So I hope that helps and that things don't get confusing, but just kind of remember, together single chromosomes, together they're a duplicated chromosome made up of sister chromatids, and then when they separate, those sister chromatids become single chromosomes. All right, have a good one.